Great. I anticipate some people will keep continuing to join, but I won't let that distract me. I'd like to kick off now. So kia ora tātou. Ngā mihi nui e koutou kua hui hui mai nei I tēnei rā. So thank you everyone for joining the hui today. Ko Libby Liggins toki ongia. My name is Libby and I will be facilitating today's presentations. Before we get started, I just want to do a karakia, so join with me if you know it. Manawan mai te Māori nuku, manawan mai te Māori rangi. Ko te Māori, kai a, hei Māori te pua. Ka pakaru mai te pō, tō mai te Māori. Aumie, huie, taikie. Thanks everyone. So I'm just going to kick off with a bit of a background. So nationally, there's a lot of interest in generating biodiversity information from environmental DNA or eDNA, as we commonly call it. The opportunity that eDNA-based biodiversity assessment methods provide continue to be honed through work like we're going to talk about today. So it's through collaboration between academic scientists, commercial partners, regional councils, and community partners, really importantly. So that's something that we want to facilitate today, and that's what today's webinar is really about, and our project, which I'll introduce shortly. So in this webinar, we're going to be hearing about some of the work that's going on nationally. There's a lot of other work going on as well. This will include important advances in technology, different kind of information gains where those gains have been applied, as well as necessary considerations of tau Māori, including Māori data sovereignty, as it relates to eDNA work in Aotearoa New Zealand. So I'm going to kick off by just introducing the project that we're part of, and I guess the hosts of this webinar are part of. So there's a number of the team here, and I'll introduce them shortly. I'll just share my screen to do that. And just a note today that we're recording this. Please feel free to email me if you want to. I think someone needs to put themselves on mute. Yeah, feel free to email me and let me know if you're not comfortable with the recording being shared. I totally understand, but for capturing the moment and the important conversations that I hope today will be had, I thought it would be a good place to start. So let me just share my screen to tell you a little bit about our project. Great, so in this work titled Stocking Aotearoa's New Zealand Molecular Library for eDNA Monitoring, what we're looking to do is to generate reference sequences for our freshwater insects, some freshwater invertebrates, and estuarine and marine fishes. So these are some of our objectives. We kicked off a few months ago, and our grand goal is to provide the sequence information for this biodiversity that's essential for being able to detect these species in an eDNA sample. So the idea is, therefore, we will be delivering a better tool for research management and communities that is more sensitive and able to detect the rare and threatened species that we're interested in, indicator taxa such as those used in the MCI, and to detect overall ecosystem change. So I'm a researcher at Massey University in Auckland, and I'm really nominally leading a bunch of people who are mutually interested in this space and wanting to do good things. And it's an Envirolink Tools collaboration funded through NBIE. Really spearheaded by the regional councils. There's several of them involved, and thank you, several of you who are joining today. Also had a lot of interactions with Department of Conservation staff, and the museum and taxonomists involved in the project are essential, as are our university partners and other research entities that are really helping as part of a wider team. So our focus on these taxonomic groups has really been informed by our the current needs of council that have been communicated to us, as well as feedback from DOC and an increased awareness and desire, I think, to understand those linkages between our freshwater and marine systems in New Zealand through really important estuarine environments. So there's a strong focus on the freshwater insects, as you can see, and the marine fishes, but we're coming together into the estuarine environment to help create a fuller picture of the biodiversity in these realms. So our approach, just quickly, is to generate these data from existing expertly identified and voucher specimens that are held in collections wherever possible, realising that many of these taxa are threatened and we don't want to over-collect. But for some species, we also hope to undertake fresh collections in collaboration with council staff, DOC staff, and mana whenua. So these will then be identified by and accessioned into collections by our taxonomist and museum teams. So from these specimens, we're looking to generate DNA sequences that are commonly used in eDNA studies to identify species in these taxonomic groups. So these are 01, 16S, and 12S. So in the case of existing collections, we are very aware that in many cases, mana whenua were probably not involved in the collection event, depending on when it happened and where it happened, and may not have knowledge of these collections existing. So this is something we want to address in this project, and I think it's a wider conversation nationally. But in these cases, our approach will be working with the museums to explicitly disclose that there is incomplete information with regard to the Māori provenance of these collections, and also that attribution is incomplete. We'll furthermore attach a biocultural notice to the individual specimens and all data that derive from them. So these recognise the rights of Māori to define the use of these data from the biodiversity and the genetic resources associated with their traditional lands and waters. So these biocultural notices can be replaced in time by iwi, hapu, runanga, with more specific labels, which will stipulate the terms of use and reuse of the sequence data, for instance. I'm only briefly touching on this here. This is part of an internationally used system, the local context system that Maui, Hudson later on in our webinar will be talking about. But just to introduce it here, that these concerns are front and centre for our project, and we're part of, I guess, a group of people nationally who are hoping to work through these issues in a more sensible way and work out some best practices. And this is one approach that we're looking at too. Okay, so our project will be run over the next 2 years, by the end of which we hope to have generated a resource that is of benefit to all of Aotearoa New Zealand. In terms of where the DNA sequence status sits and how this access will be provided as part of, an ongoing broader national discussion that we're really interested to contribute to. Just want to point out that part of us delivering these resources will also involve quantifying the limitations of eDNA in assessing these forms of biodiversity. So, for instance, we anticipate that once we have that DNA sequence data, we'll have cases where these generated DNA sequences will maybe be unable to discriminate between different species. This is a known phenomenon where some DNA sequences, or particularly short lengths of DNA sequences, do not provide the resolution required to distinguish unique species. In other cases, we anticipate we will recover genetically divergent populations. So this is in a case where we sequence more than one individual from a species from geographically distinct regions, for instance. And we may even discover some cryptic species through sequencing these multiple individuals. And we also anticipate cases where we will not be able to amplify our target DNA sequences for these species because the primers used to facilitate amplification and sequencing simply do not bind to that species DNA. So in these cases, we might find that eDNA studies using these regions of DNA will simply not be adequate for these species. And we think this is really important to know, to understand where and how eDNA studies can be most useful. So that's all I'm going to cover off today for my my work, or our work, sorry, collectively. But just to say that this is not going to be the last webinar, it's not going to be the last time you hear from us. We look forward to continuing the conversation today with these broader groups who are presenting, with people in the audience. We encourage questions and dialogue, and we plan to keep you posted on how we're going and invite you to get as involved with the project as you would like. Cool, so just get out of that. See Beck on my screen. There we go. Great. So with that, I haven't really made time for questions for me, but please feel free to use the question time and the other speakers' time to ask any questions about the broader project, and hopefully there'll be time at the end as well. But I want to next introduce Shaun Wilkinson, who's going to kick us off and talk a little bit more about those reference databases and set the scene for why the project that we're doing with Envirolink is really necessary. Thanks, Shaun.
Thanks, Libby. Can I get a thumbs up if you can hear and see me? I'm going to go yes. All right. Kia ora, everybody. My name's Shaun Wilkinson. I'm the founder and principal scientist of Wilderlab, which is a commercial eDNA processing laboratory based in Miramar, Te Motu Kairangi in Wellington. And we've been going about 3 years here now. We've got a team of around 18 people. And our main mission is to make eDNA accessible. And that includes things like designing and developing easy-to-use eDNA kits, developing consistent standardized methodologies to make sure that everybody's taking samples in the same way, and a really high throughput scalable lab workflow to make sure that we can pump out samples nice and fast and nice and cheap. And we tend to work with central and local government, tend to be our main clients, and environmental consultancies who work on behalf of them, lots of research institutes, primary sector, community groups, lots of other clients, including just ordinary people who just want to know what's in their pond or their river. And so just a real quick introduction to environmental DNA. I'm sure most of the people on this call know what it is, or approximately what it is. It's the molecular traces of living things. So things like skin and scales and hair, mucus, fecal matter that end up in the water. And this can be filtered out and taken into a lab, and the DNA can be sequenced relatively easily and cheaply these days, to create quite a scalable and cost-effective monitoring solution, which has undergone exponential growth in recent years. Certainly the number of papers that have been coming out in New Zealand and overseas have shown an exponential growth curve, and the number of samples that have been taken and used, particularly in New Zealand in the last, say, 5 to 10 years, has really shown an exponential growth curve as well. So people are using it for lots of different applications, including ecological monitoring, primarily around detecting fish and plants and insects and other critters, for biodiversity assessments, biosecurity as well, to an increasing extent for human health. So we're using it now for COVID variant detection in wastewater and bovine TB, waterborne pathogens from drinking water. And it's a great education and outreach tool. Gets people connected with their environment and understanding what's living in their waterways and what impacts human activities are having. So the way most of our clients are taking eDNA samples is using a fairly simple syringe filtration kit, where you filter a litre of water through a filter. It takes about 5 to 10 minutes per sample to send the filter back to us for analysis. And it's suitable for most applications, though there are pumps and other overnight passive samplers and other systems around as well. So once it gets back to the lab, it goes through a process, a workflow involving the extraction and purification of the DNA using the machine on the left that uses magnetic beads and magnetic rods to transfer, bind and wash DNA, take it through to a highly purified state. Then it goes into the what's called a PCR machine, which is like a little oven that cycles through different temperatures with different enzymes to create lots and lots of copies. And the key thing here is that we're just creating copies of certain tiny little regions of the genome. We're not amplifying all of the DNA in there. We're just targeting little tiny regions, which give us enough information to be able to tell what species are in there. And so we call those barcode regions, and I'll talk a bit more about those. From there, it goes into the high throughput sequencing machine, which just reads the DNA and creates these enormous files of A's and C's and T's and G's, long strings that can then be matched to a reference sequence database. In this case, we tend to use the NCBI GenBank, and a popular way of matching sequences to this database is a tool called BLAST, or Basic Local Alignment Search Tool. And so you can upload a sequence and it'll query the database and give you the top hits. In this case, it's hitting to European perch, Perca fluvialis, with 100% identity and 100% cover, and we can be fairly confident that because it's only hitting to that fish, that it is that species. And then we can produce tables of all the different species that are detected in the sample, along with their sequence counts, which are those columns on the right, and we can dress that up to make more interactive, engaging sample reports as well. So reference barcoding is super important because all of this kind of relies on this foundation of having good, high quality reference data. And it's required for lots of different things. It's really important for biodiversity assessments. If we want to be able to survey across the tree of life, we need to make sure that we've got reference barcodes for all of the different species that are in there. And so one example is some of you might have seen in the news, there was a new species of gravel, or a new gravel maggot population found on the west coast of the South Island by some dock people. And that was only because there's a particular man within dock who's really keen on Smeagol gravel maggots. He's found some before, and he wanted us to be able to detect them with the eDNA, and so he hooked us up with Te Papa, and I spent a whole afternoon in Te Papa looking through the vials of old specimens, trying to find a Smeagol gravel maggot, and thanks to Clive Roberts for that. We finally found one right at the back in a little corner, managed to get it back to the lab, do a DNA barcoding analysis on it, and from that point on we were able to detect gravel maggots in our eDNA samples. And when this one showed up in this West Coast beach about a year later, we were able to say, yes, it's definitely a Smeagol, even though it's slightly different to the one that's found in Wellington, so therefore we could be dealing with a genetically distinct population, or possibly even a different species. So despite this, we still don't really have enough reference barcodes to be able to do comprehensive marine biodiversity assessments, and the Department of Conservation's Marine Monitoring Reporting Framework has just come out for the next few years, and doesn't really include eDNA as a biodiversity assessment tool, simply because there's just not enough DNA reference data to be able to give comprehensive biodiversity survey assessments. Fine for things like biosecurity. We're targeting certain species and we know if we have the barcode or not, but if you're trying to survey across the tree of life, then you need lots of reference barcodes from different species. So biosecurity is another huge one, and we need to make sure that all the really high threat species are in that reference sequence database. And the situation we have in New Zealand at the moment is that we have a lot of freshwater macrophytes, invasive freshwater macrophytes, which unfortunately have not been reference sequenced yet. One of them was eelgrass Vallisneria spiralis, which Greater Wellington Regional Council was really keen to be able to monitor for. So they went out and found a patch and sent us a little leaf, which we then ran through the sequencer, got the reference barcode, and from that point on, we were able to detect if eelgrass was present in any samples, and sure enough, it showed up in a little creek just outside of Masterton a few months later. It was a totally unknown population. They hadn't seen it with the visual surveys, but were able to return to the site and find a newly established population of eelgrass and able to control it from there. That's another reason why reference barcoding is so important. Another one is for threatened species monitoring. So we didn't have a reference barcode for the critically endangered Clutha flathead galaxid until Dr. Richard Allibone sent us a swab sample. We were able to upload that to the database and then detect that species from now on. And since then, there have been 4 new populations of this critically endangered galaxid that have been found through eDNA that hadn't been able to be detected using historical visual surveys. Just so many miles of creeks to walk up, and they're quite cryptic, quite difficult to spot. So really cool to be able to detect those new populations and be able to instigate some protection measures to make sure that trout are not getting to them. There's no stock in the streams and things like that. Another one is for ecological health assessments. So lots of demand to be able to do an eDNA-based macroinvertebrate community index, a measure of ecological health. At the moment, we can do it, but we only have really a third of New Zealand's aquatic insects in the database, and so the molecular-based MCI is not as powerful as it could be. And as more and more of these insects get reference sequenced, we'll be able to firm up that correlation between the standard KickNet-based MCI and the molecular MCI. It's not terrible at the moment, but it'll definitely get stronger as more and more insects get reference sequenced. Another hugely powerful feature of eDNA is that we can retroactively assign species. And so we went through about 18 months where we didn't have a reference sequence for the Field blue duck. And we tried and begged and borrowed and tried to find a feather from everywhere. Finally managed to get one from someone up in the Ruahinis who'd come across I don't know what a flock of them, these are called, a paddle, and found a little feather on the side of the stream, and we managed to sequence it and get the reference barcode. And then we were able to look at all our historic data and update all those eDNA reports to show blue ducks in various different places around New Zealand, some of which people didn't know they were there. So as we scale up our eDNA monitoring, which is happening, we really need to scale up our reference barcoding efforts. And so in the peak season in summer, we process up to 500 eDNA samples a week. monitoring thousands of kilometres of waterways effectively, sequenced lots and lots of sequence variants. We've detected 3,500 insect taxa, 2,500 plants, 1,500 fish. These are not just from New Zealand, from around the world as well, and huge numbers of worms and mollusks and other things as well. But the vast majority of eDNA sequences we can't assign to a species level or anything really with more resolution than, say, an order or a family or even a phylum a lot of the time. And so having a look at the reference barcodes, the availability of those in the public reference sequence databases for the fish in New Zealand, Libby has compiled a list of 1,319 fish taxa that are either in New Zealand or that stray into New Zealand waters sometimes. And of those, we have 613 of those have the popular 16S reference sequence that we tend to use most for eDNA. Around 374, so only 28% have the MyFish 12S barcode, which is another really popular one. And 681 have no 12S or 16S barcodes, so more than half, which is a bit of a problem. Although all of the New Zealand freshwater fish currently have at least one reference barcode sequence now, so we can detect those. Birds are sort of even worse, and so less than half have the popular MyBird 12S barcode sequence, and then freshwater invertebrates. So only 260 out of 675 have the full length CO1 barcode, and just over half, just over half have the CO1 mini barcode that we tend to use for eDNA monitoring. Barcode quality is a big issue. I'll get slapped on the hand by Steve if I didn't mention this. So not all the barcodes are equal in there. Some are of dubious quality. Some of the sequencing reads have been of dubious quality. Some of the annotations, so people putting incorrect taxon identifiers against sequences and things. So that's another issue to work through as well. So a key question just to finish up is how do we fill these gaps? And I think what's tended to happen is that there's not been enough funding dedicated to barcoding projects. It's not very shiny and flashy science. It's not like that it end up in Nature or Science magazine. And so we need to really up our game in terms of the funding that's available for these barcoding projects. It typically has been done, piecemeal by people studying a particular species, and they'll upload a barcode. But what we really need is these sort of broader barcoding efforts like this project. And the BioHeritage SOVOR surveillance project as well, which Manpreet will talk about hopefully soon. Some central leadership and coordination. So at the moment, you've got lots of different people doing lots of different things. There needs to be some leadership there, I think, at a central government level. And we really need to progress the social, cultural and data sharing issues, I think, before we can really scale up the reference barcoding as well. Not a topic I will have time or ability to go into in depth here, but at the moment, genetic barcoding and hopefully at some point in the future, genome skimming, which is a really powerful and cheap way to generate lots and lots of reference data, and what's currently being done in Australia. Lots of embargoed and unpublished data that scientists are sitting on. Libby, I'm looking at you. And others on this call probably as well, which would be great to get into the public reference databases at some point, so that it can be used. And also, like I said before, there's got to be some layer of quality assurance in there to make sure that people are uploading data with the correct annotations, the correct species IDs. And so really, I think what is needed is a national reference sequence library, which hopefully Manpreet will be able to expand a little on as well. That's all I got time for. Thank you.
Thanks, Shaun. That's great. Yeah, totally guilty. I've got a lot of barcode data I'm sitting on. I'll say that the major issue is being really sure of the quality of those sequences and having that resource, that personnel to help with getting them over the line. So the Envirolink project is one way that we're going to make that happen. So wait, there'll be lots of fish DNA coming your way. Got time for one quick question for Shaun before we move on to our next speaker, who's Josh. Got one question for Shaun. We've got some compliments in the chat, but maybe we'll move on. And if you do have questions for Shaun, please feel free to ask them in the question time for the other speakers if you would like. So Josh, I'd love you to share your screen and introduce yourself. I can't see you amongst all of the other people. Great.
Kia ora koutou, everyone. Let's get my...
Looks great. Thanks, Josh.
Cool. Yeah, kia ora koutou, everyone. Josh Smith from Waikato Regional Council. Today I'm going to be talking about optimum eDNA replication in New Zealand streams, mainly focusing on freshwater fish, but we also take a look at some of the other groups. Replication. So why do we need replication in eDNA? The starting material we collect in the field can be limited and degraded, especially for rare and patchy species. As we are sampling eDNA material that is shed by organisms, it's moving downstream with the flow. And for fish species that are common to the vicinity that we're sampling, this eDNA material is easily encountered. But for other species with one or two individuals close by, or with a good number of species but a few kilometres upstream, this DNA material is patchy and random, and very rare it'll be by chance that we pick it up. But our chances increase with the more reps we take. And back in the lab, the amplification of this DNA material in low amounts can also be random. So not enough replication can lead to false negatives or non-detection. And in general, as we know, replication reduces variability and increases our confidence in our results. This all makes replication an important factor for any eDNA biodiversity study. Just got some results here to look at. This is from the Nuhaka River in the Hawke's Bay, where we've got eight reps taken and we've got 11 species present. If we look at the top 5 species, crayons down to long finial, we can see they're in all of the 8 reps. And then as we move down, we've got shortfin in 6, grey mullet in 5, bluegill 4, rainbow and red fins are in 3, and then giant bullies only in 1. Had we only taken 3 reps, we would have missed out on 3 species. And by the 6th rep, we've picked up giant bully. That just shows the importance of replication for picking everything up and reducing false negatives. Looking at the high rep inter-council study, what it involved, this was 54 sites nationwide with involving 14 regional councils, and most of the samples were collected over the 21-22 summer. 51 were analysed, where 3 were dropped because of sampling errors, ranging from first order streams right up to some of our biggest rivers in the Waikato and the Clutha, and a range of lowland sites right up to a bunch of inland sites and higher elevations. So at each site, a total of 24 reps were taken. This involves sucking up stream water with the 50 ml syringe and pushing the water through a filter, and we did this 10 times to give us a litre of water sampled. And once filtered, a preservative is added to the filter, and then from here 8 filters were randomly taken and labelled as standard, and the remaining 16 were randomly pulled back in the lab to create 8 composite samples or boosted samples. And these samples were amplified in the lab. The same amount of lysate or filtered DNA material was used for both the standard and the boosted samples. And looking at some of the results, this is for all 51 sites. The amount of eDNA material collected differs from site to site, as we can see in the graph. The lower one here is the Haast River with not much DNA compared to a site like the Clutha River here that had the highest. And this is dependent on many factors, like the amount of source DNA material available at the sampling locations, as well as many other environmental factors that will differ from site to site, like water clarity, temperature, geology, flow, nutrient levels, UV light, etc. You can see in the graph here, we've got different colours representing different groups, and fish are along the bottom, and they make up 11% of the total reads. And though we're targeting fish, as you can see in the graph here, we're getting a lot of added community information for each site. The other group in the orange here, running through the middle of the graph, these are the highest contributor to overall reads at 33.5%, and they mainly consist of unidentified sequences, which are a combination of organisms not currently in the reference database or are undescribed. And there are some interesting examples in the graph. As you can see here, this is worms here in the dark red, and at this site, Papua Nui site in the Hawke's Bay, we're getting lots of worms, and this is a lowland, highly eutrophic site. Another interesting example is a site in the Waikato, the Manganui River. We've got the highest mammal reads, and this is mainly cattle. And at this site, cattle were standing in the stream above where the sampling was taking place. Similar sort of result over here, site in Nelson with birds, and these are all these birds are 98% mallard ducks. So the main focus of the study was to establish the amount of replication needed to sample fish in New Zealand streams effectively, with the additive method comparisons of standard and boosted. And looking at the results for fish in this figure, we can see there was no significant difference between the standard and boosted method. And this is the same for all other groups we looked at for invertebrates, mammals, and birds. So there was no difference found between methods for boosted and standard. And for replication, looking at this graph for fish, we can see after one rep, we're getting 64% proportional species richness. After 3, we're getting 82, but we have a longish tail here. And then by 6 reps, we're getting close to 90% in a lot tighter fit. And this is why, discussing with all the other interested parties, we've arrived at 6 reps as what we would like to see as a standard for fish eDNA sampling. And beyond 6 reps, we get a reduction in the proportional benefit of the added reps. So overall, as we saw, there was no significant difference found between the methods of standard and boosted. So we can effectively treat all reps as standard, giving us a total of 16 reps per site. And this figure highlights the difference between groups and number of reps needed to reach a certain group richness. And as we saw in the last graph for fish, at 6 reps, we were reaching 90%. If we want 90% richness for mammals and MCI invertebrates, we're looking at close to 10 reps. And for birds, to get that 90%, we're looking at around 13. And there are a number of reasons why fish do considerably better than the other groups. There's less total species for fish compared with something like MCI invertebrates. And obviously with mammals and birds being largely terrestrial, there's also some differences in markers between the groups. Terrestrially linked information from birds and mammals and plants, as we've heard from Shaun, aids in conservation and biosecurity efforts. And with this study, we picked up a lot of rare bird species, and we also picked up plenty of mammalian pest species like mustelids, rats, and possums. Putting this into context for fish, how well does eDNA do against traditional methods? Electric fishing is the main method used for fish monitoring in New Zealand, and at 6 of the sites in this study, as soon as the eDNA sampling was completed, we conducted an electric fishing survey. We need to note that there's a big difference between electric fishing and eDNA. For electric fishing, we're sampling 150 metre length of stream, whereas eDNA, the distance will be well beyond this. But looking at the results, we can see this is eDNA 6 reps taken, and we got a 31% increase in species richness for eDNA. For 4 of the sites in the Waikato, these are our reference sites, we have data, fish data, for 10 plus years, and when we look at that data compared with eDNA, we get an increase of 14.5% richness with eDNA. And for all the extra species that were found with eDNA, they make sense. And with 2 species were missed with eDNA. These are both rare and picked up with electric fishing, showing that very rare species, even with eDNA, may be hard to find. And there's a paper put out last year, early last year. It's in the New Zealand Journal of Zoology, if you wanted to look at a bit more on fish and eDNA. Thank you.
Thanks, Josh. And sorry, I totally didn't introduce you. So in case you missed it, Josh is a monitoring scientist at the Waikato Regional Council, and the work that he presented was part of a master's project at the University of Waikato. So we've got some time for questions for Josh. Has anyone got something they'd like to pipe up and ask? I have a question. So based on your master's work and working in the Regional Council, Josh, what you found in terms of the number of replicates, is that something that's now put into practice within Council?
It is, yeah. So for particular sites where we want SOE information on, we'll take 6 reps.
And is it within just your council, or are all councils now following what you've determined to be best practice with the eDNA?
A lot of other councils are doing the same as well. So most of the councils were involved in the study, and the results we came up with were put out to them and discussed with them, and we've sort of arrived at 6 reps for the A standard of fish sampling with eDNA.
Yeah, cool. That's really neat. Really nice study. Thank you for sharing that. Any other questions for Josh or for Shaun from his earlier talk?
Yep, I got a question. Kia ora, my name is Morgan. I'm at in biosecurity at Environment Canterbury. I was just wondering, Josh and anyone else, has there been replicate testing done for eDNA for plants, whether that be sort of native or pest plants or whatever?
That might be something that Shaun may want to answer there.
Yeah, we haven't actually done the replicate validation for plants yet. There's the total plant sort of biodiversity kind of assessment, or if you're looking at species accumulation curves like what Josh was just showing for the insects and bugs and things, will be a really flat curve. So it'll just like kind of gradually go up and still be in the linear phase after I imagine 16 reps, just because there's just so many terrestrial plants, so many species that are coming into it. But I think this is a slightly different question, because if you're just interested in, say, half a dozen or a dozen key species, your hornworts and your eelgrasses and that sort of thing, I think that's something there's a bit of a hole in the knowledge of how many reps do we need to be able to detect those key species with those sort of more general metabarcode and primers. So yeah, that's a good point, and probably something that we want to attack at some point in the future. Thanks, Morgan. Awesome,
thank you.
Yeah, thanks, Morgan and Shaun. There is a couple of questions in the chat there. So GJ is wondering, for the replication level needed, is there consensus between sites and environments? So that's a question for Josh.
No, so it's so every site's going to vary depending on environmental factors. So it's very hard to compare site to site, but in general, if you're wanting to get a biodiversity metric from eDNA for fish, we recommend 6 reps.
And that's for riverine environments only, really, isn't it? So that could be probably more for lakes, I imagine, and other environments where the DNA isn't sort of just coming at you on a constant lovely conveyor belt, and where DNA can be localized and patchy if things are living in sort of one microhabitat as opposed to another. So I think important to notice that this 6 rep recommendation is really only applicable for riverine habitats at this stage.
Great. Thanks, Josh and Shaun. That's a nice segue to our next speaker. So just before I introduce Susie, I just want to note that there is a bit of chat going on in the chat. So do open it up and have a read and ask questions. There's a question there for you, Josh, if you want to get to that from Manpreet about rare species. I look forward to the answer. Cool. So our next speaker is Susie Wood. So she's a freshwater scientist and molecular ecologist at the Cawthorne Institute, and she co-leads the Lakes 380 project, which you may have heard about, and she's going to talk about today. And this is in conjunction with DNA science as well. So thanks, Susie. Take it away.
Tēnā koutou katoa. Thank you, Libby, for the introduction. Everybody can hear and see my screen okay? Yep, excellent. So yeah, as Libby said, I'm a freshwater scientist, molecular ecologist, and I want to talk today about a project known as our Lakes Health Past, Present and Future, or Lakes 380, that we've been working on for the, I guess, the last 4 or so years. And the aim of this project is to assess the current and historic health of lakes in Aotearoa. Oops, here we go. And so, yeah, in the first 2 or 3 years of the project, we put a huge effort into sampling lakes. And I guess most importantly, what we aimed to do was collect samples from a highly representative site selection of lakes across New Zealand. So all sorts of different gradients from altitude to land use to size and depth. And so it is the most highly representative lake sample set in Aotearoa. And we were successful at sampling around about 10% of our 3,800 lakes. And so, yeah, environmental DNA is a really important part of our project. We've both developed and validated our own methods, as well as validated and applied other methods. And as you can see from this image here, we're primarily interested in aquatic organisms, but we've become increasingly interested in the terrestrial signal that we can also pick up. We primarily have worked on sediments, but we also collect water samples as well. And so when we're visiting our lakes, we're collecting a whole suite of samples for many things, not only for environmental DNA, but a whole raft of other samples as well. But when we talk about DNA, we're collecting surface water and sediment samples to characterise the current health and biodiversity of lakes. And we're also collecting sediment cores. So year by year, sediment it's laid down into a lake, and it can act like a storybook or a history book back in time. If we analyse those different layers, we can understand whether our lakes have changed and how and why they've changed. And so I'm just going to give you a quick snapshot of some of the environmental DNA projects that have been underway as part of this work. So focusing on some of the work we've done around current health and biodiversity. So one of the challenges when we think about understanding current lake water quality or trophic state is that our current approach involves taking a boat out into the centre of a lake and collecting a water sample and measuring the algae and nutrient concentrations. And while that generates a huge amount of really valuable data, it's time consuming and limits the number of lakes that we can monitor. And so our first challenge to overcome was to develop an eDNA-based method that allowed us to rapidly estimate lake water quality. And we achieved this using surface sediment samples. So surface sediment are a wonderful indicator of time, and characterising the bacterial communities using environmental DNA. And so we're able to use statistical approaches to find indicator bacteria and then develop what we call a surface bacterial trophic index. And we validated this method using data from 100 monitored lakes with long-term monitoring records, and compared this to the data that we generated using our surface bacterial index. And these samples were collected in triplicate in the field and analysed in triplicate in the laboratory. So we're really keen to develop an incredibly robust method. And this graph here shows the relationship between the traditional trophic level index and our surface bacterial index. So we're fairly confident and happy that we've got a really nice, rapid, robust way of estimating trophic level. And this can be achieved by taking a single point in time sample from the sediment. And we've just taken this a step further recently and used machine learning or statistical approaches to take the data that we've generated from our 300 lakes and apply this to all lakes in Aotearoa, New Zealand. And so this is what this graphic here shows, this this data, and we've developed a nice app that will be launching soon where you can dive in and have a look at this in more detail. And just to give you an example of how this information has been used at a regional scale, so there's a number of regional councils now that are now using this information to help with the implementation of the National Policy Statement for Freshwater Management, and able to use this information to identify lakes their action plans or restoration plans might need to be implemented. So a great example of environmental DNA being used to aid with environmental management. Okay, so as we travelled around visiting or sampling the lakes, we had a huge amount of feedback that there was a lot of interest, particularly in some of the tionga species, in particular kakahi and tuna, and we were often asked if they were present in the lakes and or whether they had been present. And so we put a huge focus on kakahi and tuna and developed really sensitive, targeted, specific methods, as opposed to the meta-barcoding methods that characterise entire communities. And these methods go in and just target, in the case of kakahi, 3 different species or 2 different species. And this makes them incredibly sensitive methods. So once we'd developed those, we were really happy that we had great techniques developed, but we wanted to think about making sure that we were robustly applying these methods to modern or current day sampling of lakes. And so we asked ourselves the question, how many samples do we need to collect to be sure, in this case, that we've got kakahi in our lakes? And this was a study that was carried out in Aotearoa or the Ashburton Lakes region, in collaboration with Environment Canterbury, Department of Conservation, and NIWA. And so we actually worked on 7 lakes. There's a great set of lakes there that within a small geographic zone differ in their depth and their size and their trophic levels. So it's an excellent place to carry out the study. And as well as in collecting environmental DNA samples, we had the NIWA diving team out there actually measuring the abundance using diver surveys. So I'm just going to show one result from this study. And when we combine all the data together, it allows us to use a statistical approach known as occupancy modelling. And this is a highly accurate way of estimating how many samples you need to collect from a lake with high levels of certainty to be able to detect kakahi. So this is the sort of final output of that model, and how we can use this is essentially if you're fairly confident you've got kakahi in your lake, then you'd only need to take 4 water samples, this is, from that lake to be 90% confident that you've got kakahi, or whether you do or don't have kakahi in your lake. Sorry, whether you do have kakahi in your lake. But if you're much less certain of, if you've got no idea if you've got kakahi in your lake, actually the number of samples that you may have to take is much, much higher, can be up to 40 or 50 samples. So yeah, I guess just a really important message around understanding what, this problem with eDNA. If we don't get a positive result, what does that mean? And actually we think, the need to do this sort of work, and particularly in lake environments, is really, really important. And so a similar study that we did as part of the project in 3 lowland lakes, this time targeting non-native fish, so the rudd and European perch. And in this project, we asked the question, does it matter where we get our sample in the lakes? So does it matter if it's on the edge of the lake or in the middle of the lake? And we also asked whether it was more important to take water or sediment samples, which would increase the likelihood of detecting these fish. And so in summary, essentially we found that it made no difference where you collected the sample in the study. It could be either at the shore or in the middle of the lake. It made no difference. But what we found was that every lake was slightly different in terms of whether water or sediment samples gave you the highest likelihood of detecting the different species. And it also differed between species a bit. So in Lake Paunui, we were much more likely to detect perch if we collected and analysed sediment samples. In Waitawa, it was much more likely to pick up both perch and rudd in the water. And in fact, it was hard to find their DNA in the sediment, and this is to do with we think to do with the geochemistry of the sediment, and the DNA doesn't bind well to it in that lake. In Tomarata, there's hardly any perch anyway, but for rudd it was much better. We had a higher likelihood of detecting rudd if you used sediment samples. So just to show you, particularly in lakes, it's not always clear-cut how many samples you should take and where you should sample and what sort of sample you should take. And it's really important to understand the organism that you're working on and the system that you're working on. Cool. So now I'm going to take you on a trip back in time and show you some of the data that's coming out of our sediment cores and just how our lakes are changing over time. And so the first lake I'm going to start with is Lake Ponui, which is, you saw an image of it just before. It's a small lake in the Wairarapa region. And what we've got here is the sediment core, and this is spanning 1,000 years of time. And this data here is that bacterial index that I introduced you to that we'd applied across the surface of our lakes, but now we're using that same index back in time. And so prior to human arrival in this region and also during the period of Maori settlement, this lake was in a good or reasonably good condition. But post-European arrival, our eDNA data shows the reasonably rapid decline in water quality, and this lake is now a hypereutrophic lake. And this kind of information is extremely invaluable when you're going about setting restoration targets. It gives you some accurate indications of where you might set, how you might go about setting informed restoration targets. So I want to stick with Ponui now and show you a little bit more of the data from there and also give you some insights into why this lake has declined. And so as I mentioned earlier, one of the questions that we got asked a lot as we travelled around were kakahi and tuna present in my lake. And so this is some data again from the Lake Ponui core. And what we had to do was one of our great PhDs, Georgia Thompson-Lang, who developed a method to extract DNA from these ancient sediments, which proved much harder than we thought, but she achieved this, and we've now been able to detect many of these important species back in time. So we can see here that over the last number of years, 700 odd years, that we had both kakahi and tuna in this lake. Just wanted to highlight there are some places where we don't detect them. This certainly doesn't mean that they weren't there, but just really highlights the patchy nature of environmental DNA, in particular fish in our lake environments. So it's absence of a DNA signal doesn't necessarily mean that organism wasn't present. And I would just stress that it's a wonderful technique, environmental DNA, but we also have to be aware of those caveats and make sure we interpret our data with those in mind. So one of the really interesting things was that when we applied our assay for perch, we found that we could detect perch back down to about 1870. And so prior to this study, we didn't actually know when perch were introduced into Ponui. There were no written or documented records of this. So this has given us a really nice point in time when perch were introduced, which then allows us to look at how the community has responded since perch introduction. And so just to show you some examples, these are GAMS plots, and essentially where the line is red, that means there's a significant change in the community. And what I've done here is just pick 3 different communities that we monitored using environmental DNA in the sediment cores. So entire micro-eukaryotic communities, so the zooplankton, rotifers, various different algae, bacterial communities, which we know are wonderful indicators of change, and macrophytes as well, so we can track them back through time. So perch were introduced around about 1870, and then we see these different communities starting to change significantly post perch. And we know in this region that while there was a little bit of land use change, it wasn't very significant, and we really think that perch were the main drivers of change and decline in water quality in this lake. So some really powerful examples of how environmental DNA can be applied to these samples to really give us a good indication of why our lakes are in their current condition. Yeah, and so just to finish off some lessons that we've learned from using eDNA and developing and applying eDNA in our study is that it's really important to know what your question or your purpose of applying eDNA in lakes is. And so are you interested in a really highly sensitive specific method to detect one species, or are you more interested broadly in community composition? We've learnt that all study organisms aren't equal. Certainly, you couldn't take a water sample for which works really well for kākahi. We know that it doesn't work very well for tuna and some of the other fish species. It's really important to understand the system. So a highly eutrophic small lake will be very different from a large deep lake in terms of number of samples and how you should go about sampling them. And yeah, really importantly, again, it's a wonderful technique, but we need to make sure we interpret our results with a good degree of understanding. So when you don't detect something that you thought was there, or you didn't know if it was there, what does that mean and how certain can you be about that? And yeah, just to finish off, to thank the amazing Lake Strategy team, who are a group of really passionate and dynamic researchers. So yeah, and thank you very much for your time.
Thanks very much, Susie. What a huge body of work, and really nice to see that, a big part of it was actually quantifying those caveats as well and qualifying the methods appropriately. We're running a little bit over time, so I'm sure you've got heaps of questions for Susie. Please put them in the chat, and hopefully we'll have time to ask them in person. But I'm going to move straight on to our next speaker, who's Manpreet Dhami. So Manpreet's a senior scientist in molecular ecology at Manaaki, Whenua. She leads eDNA technologies for surveillance systems within the state-of-the-art surveillance technologies program, which is part of the National Science Challenge Biological Heritage. Take it away, Manpreet.
I should unmute myself first. Thanks, Libby. I'll just kick right in, and I think my talk is just about an overview of the project so I can catch up the time. Well, kia ora koutou katoa, ko Manpreet Dhami tōku ingoa, ke Manaaki Whenua, aho e mahiana. Hello everyone, I'm Manpreet Dhami, and I'm a senior scientist at Manaaki Whenua Landscape Research. And as I said earlier, today I'm just going to give an overview of the ongoing research within the state-of-the-art surveillance project, especially the eDNA or mainly terrestrial eDNA part of the project. As the title suggests, I will talk today why and how we are trapping DNA for biosecurity outcomes. As is the case for all of National Science Challenge projects, I'm representing a group of dedicated researchers and collaborators from various organizations, some of whom are on this call today. So typically I give a broad overview of how eDNA has been used in biomonitoring, but today I feel like we've already seen some really excellent examples of how this is done, and especially how it's done in Aotearoa. But just to cover off the slide, as sequencing is becoming faster and more accessible, we can now collect hundreds and thousands, even tens of thousands of samples. And we're starting to build maps of where these species are located. And as we do more and more of it, our ability to predict and project where species are present is getting better. For example, here's a map of all of earthworms that was globally that was published in 2019. Here's another map looking at where all the fungi are present that came even before in 2014. And closer to home, and as we've seen for the examples, here are some lists of species that we can detect using environmental DNA. This is an example from Hudson, which is one of the larger projects that went around in New Zealand around a while ago, actually in 2015. So generally to conclude from here is that we can see that environmental DNA approaches can tell us about species, about where species are present in an environment, and now they can be applied at very large scales, especially as we just saw for the Lakes 380 program as well. And these developments have primarily been very promising in the aquatic eDNA space, where the environment is nice and flowing and accumulates in a nearly uniform manner. And we've seen through increased research as well as applications coming out of eDNA as well as eRNA space in the aquatic environment that this is a very promising avenue. And as also just Susie has been talking about, there are some things to consider, but overall, I think we can gain a lot of insight on biodiversity monitoring for fish and invertebrates, and sometimes even terrestrial species that interact with water. There are also many proven applications for diagnostics and biosecurity. including invertebrates and vertebrate pests, and sometimes even new to New Zealand species that have just arrived. And I think that one of the cases that gets us very excited is recently, as part of routine testing, I think Shaun identified fall armyworm in New Zealand very shortly after it had arrived, which was really exciting to see the diagnostic potential of these techniques. But aquatic DNA doesn't encompass everything, especially for strictly terrestrial species, which may never encounter water or be blown into a river. The ability to detect these certain terrestrial species can be quite limited. And a vast majority of biosecurity threats that we face in New Zealand remain species that are primarily with a terrestrial lifestyle. So what are alternative ways that we can utilize to improve our ability to detect some of these problematic terrestrial threats? So terrestrial environments, as I mentioned earlier, don't typically lend themselves very well to DNA-based detection. They don't have this uniform flowing property to them. DNA degrades unevenly in soil, and for small or rare species, you might need very large sample sizes to increase diagnostic confidence, all of which really compounds technical challenges. In response, scientists have been developing cunning ways of aggregating DNA. So this is a bit more like, a bit adjacent to eDNA proper. I think purists might even argue that this isn't an eDNA type approach, but it is still collecting DNA signatures of various species. So for our discussion today, we can pull it with other eDNA projects. But basically, what I'm talking about is utilizing an alternative method to accumulate DNA of species that are primarily terrestrial. For example, this vacuum cleaner here can double up as a DNA aggregation device, and has been actually applied successfully to detect copra beetle in shipping containers in Australia. And similarly, wastewater testing for COVID can also be thought of as an aggregation method where wastewater from terrestrial households was being collected to detect human-associated COVID-19 pathogen. And similar research has also looked at the ability of bees and even flowers to be locations where insect DNA might accumulate, or the DNA of pathogens that bees might move around would accumulate as well in their pollen baskets, and have been used to detect things like myrtle rust and various weed species. But among key terrestrial biosecurity threats for New Zealand are actually insects. In fact, around 2/3 of listed terrestrial species that are a problem for New Zealand are insects, which is quite a vast number. So these are all just examples of DNA aggregation devices. And for collecting insects, a widely used method is an insect trap, right? Anyone who studied insect biodiversity in university or later on in their life has put up an insect trap at some point. With the right preservative and a generalized attraction cue, such as a UV light, a simple light trap can become a pretty accurate DNA aggregation device for insects. And a network of such devices are already deployed across New Zealand, especially at high-risk surveillance sites, such as ports or transitional facilities where we have a lot of traffic for introduced species coming into the country. And as part of one such program, we've teamed up with the Port of Tauranga, where our collaborators, Carl Warder and in the photo here from Sion, have been sorting through hundreds to thousands of insects, sometimes including new to New Zealand species that appear at the ports. These collections now are doubling up as a starting material for our project, effectively an insect soup for DNA-based diagnostics. As part of this project, we're trying to uncover, again, very similar questions to what the aquatic folks have been trying to answer as well. How often do we need to sample? What time of the year? And in total, how many samples give us a really good idea of the full gamut of species arriving at or present at one of New Zealand's busiest ports? And as these samples are starting to come in, one of the important questions immediately is becoming, how do we separate the contribution of large-bodied insects like this chunky moth here from some of the smaller insects like this ant or a fly, whatever. I can't tell from this image, but that biomass associated variability in the samples. And again, we saw from Josh's talk that some of the rare species that are hard to detect is possibly because of that biomass issue. And again, a similar issue for us as well in these insect trap samples. And to do that, we've been kind of working through some of these technical innovations to try and develop a way that we could attribute the biomass proportional DNA to all of our samples. And specifically, this is work that Preeti Panda, who's one of the in our team at Plant and Food, has been developing a simple size-based filtering method to separate and process large and small insects on their own, before combining their DNA in different proportions for the diagnostic pipelines downstream. She's been using 4 different gene regions, and again, these are similar to the types of regions that have been used for inverts elsewhere. And by doing so, she's testing whether we can streamline this process to make it simpler and effective, and also which of the 4 regions or a combination of these gene regions may be optimal for species diagnostics in these high biomass samples. Once we have all the DNA in hand, the next challenge obviously becomes that of species identification. And I know Shaun spoke briefly about that just before, so I won't go into too much. But we all agree, I think, generally that the first step around that is to have really good reference sequences, right? So what we've done to get that in our end of the project is basically we've developed a rapid DNA database generation pipeline using Illumina high throughput next generation sequencing. And basically what we're currently doing, and this work is actually currently in review, almost nearly done at Molecular Ecology Resources, but the preprint is already available. I'm happy to put that in the chat, but basically it's a rapid method to generate lots and lots of barcodes at the same time from locally collected references. So the insects that Carl is sorting out of the traps, we can take those insects that have been identified and quickly build a local reference database, in fact, quite cheaply because we're utilizing Illumina technology. So while we're getting better and better with the technology of generating these sequencing, it's still nigh impossible to sequence everything. So we also need to be good at filling the gap where we don't have a sequence. And part of this effort involves Shaun, and he, I think, mentioned a little bit about it earlier, that what we're also doing is, again, curating that national DNA reference, national data set of available sequences, also identifying what things are missing, and also getting better at developing improved algorithms for species matching when there is a sequence that's completely missing. So combining all this together, I think we have a pretty good pipeline in mind that is currently being tested as we have now received the data from our first year of sampling. And we've processed the samples, sequenced everything from Port of Tauranga samples from the spring, summer 2022. And we're currently analyzing these data against a locally curated database of sequences from Port of Tauranga, plus the sequences that are currently available on NCBI. And the idea here is to assess that initial collection efforts. How well did they go? Can we learn something from those initial collection efforts to now apply in this year's upcoming sampling, which is approaching very fast. We're already in October. That can be applied to improve any of the ways that we can get better at identifying these insect species. So the grand goal of this project is to develop a workflow that can be adopted across New Zealand by agencies such as MPI, as well as Te Tira Whakamataki, DOC, Regional Counting. Many people on this call, I believe, would be interested in something like this. By solving some of the challenges to operationalizing this insect trap method, both for biosecurity in this particular instance, but in the long run for biomonitoring. So over the next 12 months, we're going to be working on 3 main things. First, we're going to continue developing this operational light trap approach and the associated DNA diagnostic pipeline as an example for best practice use. And we've got the data for this year, which will inform next year's sampling collection, and we'll continue to iteratively improve this method. We will seek interest from our stakeholder for capability development for using this workflow as well. We're offering student scholarships, particularly to increase Maori participation in biosecurity surveillance. If your organization sees a need for DNA-based surveillance for new to region or new to New Zealand pest, an insect pest, please get in touch. We'd love to hear from you again and what can we be developing for helping out our stakeholders next. And I think this keeps coming up in all of eDNA technology-based conversation, and it is one of the most important points, is the reference database development. It is absolutely critical, as has been mentioned already multiple times today. Generating this data to represent our native species requires bringing together all interested parties, and that's where I think we have to do a good job to make sure that we can continue to develop these technologies for use in perpetuity. We are currently developing an additional piece of work alongside the National Science Challenge project, specifically to talk with relevant researchers as well as Māori partners to scope out a process for a collective buy-in for generating this kind of a database, to do it at a national scale. And lastly, to use these tools, we also need social and cultural license. People can be a bit skeptical or scared of molecular things. The idea of, DNA surveillance in your backyard does not sound very appealing. So we need to socialize these ideas, and Shaun and others are doing that kind of work to bring in kids and to bring in communities to try and develop the community engagement opportunities, and to kind of, flow this bit of information and knowledge out into the wider public. There's a lot of promising development, as we can see in even the few talks that we've had today in this space, and we're excited by the prospect of how this technology will transform our ability to monitor and resist the biosecurity threats to our biodiversity and production sectors. And I think that was pretty much the overview of the project. And yeah, thank you for listening, and happy to take questions.
Yeah, thank you very much, Manpreet. I'm just conscious of time, so please, if you have questions, fire them into the chat, or we'll hit up Manpreet later if we've got time at the end. So our final speaker today, there was a nice transition there from Manpreet's talk, is Maui Hudson. So Maui's the director of the Te Kotahi Research Institute at the University of Waikato and co-director of Local Context. Maui, I'll let you introduce what you're going to be talking about today.
Yeah, thanks. Kia ora, everyone. My name is Maui Hudson, no te whakatoa here, and I've been fortunate enough to work in with Libby on development of bicultural labels. And so I think what's been really interesting today, and I've been involved with putting together guidelines around genomics and research projects for a number of years, is what we've seen from the presentations, I think, is the potential value of these sort of technologies to help us with sort of monitoring activities and management activities in the environment. At the same time as that's been happening, though, and as Manpreet sort of highlighted, is there's a need to work in partnership with Māori and iwi around how that comes about. And so as all this data is being generated, how is it being used and what it's being used for becomes something of interest to Māori groups. So, just want to talk a little bit about what we've been doing in the context of local context, and Libby mentioned it at the beginning, and just some of the ways that we're thinking about recognising Māori interests in data that can be generated through a range of projects, but in this context, eDNA projects. And so part of this, I guess the background for this is the thinking that's been happening around Indigenous data sovereignty and Māori data sovereignty. And it's about what sorts of rights and interests people have in relation to the data that's generated. So when Manpreet was talking about, having to deal with issues around social license and cultural license, really about what people's level of comfort around how that data is being used by others. And so we've got these elements under the Māori data sovereignty umbrella around data for governance. So how can iwi and other groups access that data to help support their decision making? And then as that data is being made generally available to everyone, what are the instances where there should be some form of governance in relation to that data? And so as different standards are being created to help us deal with the use of data, we've certainly been talking about how data can be made fair and how the FAIR principles support an increasing move towards openness in relation to how data is shared. And there's also been the CARE principles for Indigenous data governance that were created to focus more on the people and the purpose for which the data was being used, rather than just the characteristics of the data itself. And so these become sort of framing, framing elements which can be thought of as we start thinking about how these Māori rights might be addressed. And so in the context of these conversations that have been going on around Māori data sovereignty, but also treaty interests and treaty rights, there's these sort of common elements that start to emerge around how groups being acknowledged, how their contributions being attributed, and in some places that could relate to authorship as well. And as that recognition of those rights and interest in that information and data emerge, how do they translate into discussions about access and ultimately authority in relation to future uses? So in our sort of academic context, one of the ways in which we do that is, the use of copyright, different sorts of IP mechanisms which allow us to not just assert a form of ownership, but ensure that when our work is then being reused, that we're cited as authors or we're cited as researchers, or there's that sort of recognition as things move along. By the same token, as we've moved towards open data and started to use Creative Commons licenses as a way of enabling that sharing and use, but it still maintains that recognition of that contribution. And you can only get a Creative Commons license if you've got the copyright in the first place. And this is often one of the areas where Māori communities, iwi, are upset that they're not recognised through that process in the same sort of way, and so they're often not the IP holders, and it sort of starts to exclude them from this form of recognition. And that's where the labels have come into play, or the notices and the labels, to try and ensure that Indigenous cultural authority or information about it is maintained with the record. And so as Libby had highlighted, there's notices that have been put in place. These are particular tools for researchers and institutions so that they can indicate to communities that they're open to collaborate, open to engaging with this sort of activity, and that they can just make disclosures on the records about interests which they know that there's an interest there, either in relation to traditional knowledge or biocultural resources, but they're not necessarily the right people to say what those interests are. So we've had examples through the development of the system where it's been applied in different institutions. This is an image archive in Canada, and so this is one of the most used sort of image archives in North America, and they have a notice there that just says, as an institution, they're open to collaborate with communities around ensuring their cultural authority is recognised and on that material. Similarly, this is Geome, so this is a geological and genomic metadata database. Libby works with them quite closely, but they also have the open to collaborate notice on that as an example of it being used in a sort of a scientific context. And just as an example of what a biocultural notice looks like on an actual record, this is some work that was done by Plant and Food Research, sequencing a bilberry genome. And they put this in place because the bilberry in Scandinavia, and they've got the Indigenous Sami population that have interests around that material. So the notices were something that were generated to ensure that institutions can really start work around developing or making changes within their infrastructures. But the purpose is to ensure that the labels, whether the traditional knowledge labels, are in place. And so this initiative started about 12 years ago, really trying to deal with this issue around recognition of iwi interest or Indigenous community interest in records. They're digital markers that can sit in the record. The labels themselves are customized. And you can see that here. The label and the name remain consistent so that you can recognize them across, between different communities, but also on different institutions. But the text that sits alongside them is customized by the community. And here you have, this is an example of the Scowlitz community, which is an Indigenous community in Canada. And they've used these labels on a website for Virtual Museums Canada, which talks about their community. And so this is just giving users or next users or people that are accessing that information an indication about who they are as a people. And so what the labels themselves start to communicate, and there's 20 traditional knowledge labels, there's also 10 biocultural labels. Some of them are focused on provenance and kind of pointing back to the community, like the attribution label, or maybe saying that the hapu or the whānau are the ones that have the authority to speak about that material. There's protocols, traditional protocols for sharing, may have been restricted on gender lines or at certain times of the year, or it just has cultural sensitivity or sacredness around it. And also there's labels that relate to permissions. So whether that's, this can be used for outreach, research purposes, we're open to collaboration or commercialization. All of these things can be indicated through the use of the labels. And so the thing that's probably relevant in this context is really the biocultural labels. And these being developed particularly for genetic resources. And so in a similar sort of situation with traditional knowledge where that IP doesn't apply directly to it. And so you can't necessarily get IP directly over the genetic resource, but you can get copyright in relation to material that's been developed. So the biocultural labels were developed through a project funded here in New Zealand, and we have an Aotearoa Biocultural Labels Working Group, which Libby was a part of, and that was a project that started about 4 years ago. And it's generated the labels, and now they've started to be used in a range of different contexts. One here is the main eDNA project. So this is, they've agreed that it's important to recognise Indigenous interest as they're collecting samples from off Indigenous territories. And so they've reflected that on their website, saying that they're open to collaborate, having initiated discussions with the 4 different Indigenous communities in the state of Maine. They've been looking at how they can also reflect that in the collection of data, and they use Survey 123 as their sampling survey mechanism. And they're just also reflecting that for the various different students and staff members that will be collecting that data over the coming years, that they need to be sort of respectful as well. And we've been starting to talk with them about how that information would then start in the survey form through the data collection phase, but then start to become a part of the data flow as it gets into the metadata pipelines, and starting to think about entity relationships and how local context project ID, which is associated with the labels that are put in place, can then be connected into the other IDs and information flows which they have for that work. Just want to tell you, so a lot of the work with main eDNA has really been about some of the sort of internal aspects of how that information might be collected and managed. And then this project is just showing you where things have got to with Manaaki Whenua, and particularly Aaron Wilton and Holden Hohaia, that have been thinking about how to integrate the labels of the local context hub into their live systematics collection data. So this is their systematics collections data. If you go onto this site, it has over 600,000 records. That's highlighted there. Every single one of the records now has a biocultural notice in place. You can see that reflected there. And if I just sneak back here on the left-hand side, you can see there's a module on the side called Native Lands. You can go into that and it will, you can filter these data sets by tribal boundaries. If you filter it by tribal boundary, like in this instance, and this is for Whakatohea, so this is my community, you end up with 1,200 records. They created a project around the biological specimens housed at Manaaki, Whenua, within Aotearoa, and you see that there. This is the Whakatohea account on the local context hub. We received the project. We received this notification about the project and had a biocultural notice on it, and invited us to apply labels to it. And you see there the labels which we were interested in applying. One's a provenance label, one's a research use label that we're okay for it to be used for research purposes, and then also labels that say that we're open to collaboration and open to commercialization. So if people decide to engage with the records, and this is what it looks like on all those 1,200 records, our labels are sitting there. If they want to use it for any particular thing, we're open to them engaging with us. So this is sort of, our really first example of this in place, and it's really great what they've been able to pull together. The other thing I thought I'd quickly just point out is also been engaging with the European Reference Genome Atlas, just in the context of putting together DNA archives and reference atlases. This is one that's happening up there. They've also been thinking about all the different sorts of metadata required, and doing that in a way that's aligned with FAIR and CARE. They have a metadata manifest which highlights a whole range of things which need to be collected, including ethics permits, CITES permits, and just been moving towards also including the TK or biocultural labels on there. One thing I think it's also important to recognise is that not all the data that might be relevant can be captured within the labels, and it's often necessary to build other fields for other sorts of Indigenous metadata, which iwi or Māori groups might be interested in including in these kinds of collections. So anyone interested in talking about learning more about this can contact our program lead, Janet Hamilton-Pierce, at the email there. And we have a range of sort of working groups that are talking through this as we start to build it out with different communities and different institutions. So, kia ora, Libby. Hopefully that wasn't too long.
Kia ora, Maui. That was great. So thanks everyone who is speaking today. We've got time for one quick question if someone would like before closing. I just don't want to keep people longer than they signed up for. Is anyone game for a last question? It's clear to me that we're going to have to reconvene to have a discussion time once we have time to reflect on these presentations, because I think there's a lot of momentum, a lot of parallel work happening, and I think there's going to be a lot of value in sharing those ideas and best practices in terms of, assuring the quality of our reference databases, also the methods by which we're analysing our data and making sure it's fit for purpose, as well as the cultural and social license aspects as well. Okay, so there's been a call. Oh, jump in, Andy, go for it.
So I was just it might not be a quick question, but Maui, so we want to get a voucher. There's a bug flying around that we've noticed. Is it okay to take a sample of that bug before you've spoken to anyone? Or should we really just hold off doing anything like that till we've got our ducks in a row?
Yes, I don't know if that's a short question. I think it's a yes, no, maybe one. I mean, I think people sample a whole lot of things when they need to, but just like things that happen with COVID, if you have a biosecurity incursion, people are going to want to do that sort of sampling earlier on. Sort of one of the reasons why we've developed the notices. Much of the historic collections haven't been collected with the sorts of permissions we might expect now. So we can still do the work, apply the notice, knowing that there's an opportunity for communities to talk about their interest and add it to the record later. I think that sort of process has to be balanced with the sort of relationships that you're creating with those communities anyway. So I don't think they're independent of each other, but they can happen alongside each other in different forms. I think a lot of times there'll be mostly, the relationship is being developed before the labels get put in place or the notices are used, but in other instances it's going to be, I think the other way around.
Oh, thank you. Yeah, I know it's complicated. It was just a lot of us are struggling with what foot to put forward first, and yeah, it'll be good to get some more talk and guidance around that, so that people do things right and feel safe in what they're doing.
Thanks. Yeah, those are good points. Thanks, Andy. And yeah, we haven't resolved it. We probably will take some time to do that. But I think the conversations and the journey is really important, and I think we're all on that now. We're talking about it. Cool. So there's some things going on in the chat. I'm going to stay online if people want to keep chatting and using the chat, and if there's things to follow up. But please feel free to drop off now as soon as Maui has closed the meeting officially. So thanks, Maui.
Really, kia ora. Okay. Well, kia ora everyone. Thanks for coming along. Thanks, Libby, for putting this together. This hei whakakapi ngā kōrero mō tēnei wāhanga, kia tau ki tātou katoa, te ata whai tō tātou ariki kraiti, te aroha te atua me te whiwhinga te itanga ki te wairua tapu ake āmene. I'll hang around for a bit too if people have questions.
Got it. Thanks for that. And thanks everyone for joining. We'll keep in touch. Lots going on in this space.
Richard Harmon had his hand up there just while we're getting close to close. I'm not sure, Richard, if you're still there.
Kia ora, Andy. Yeah. I just I put my hand down because I thought, this has probably already been said, but I was interested to know from Maui how you resolve contentions between hapu and iwi on ownership.
Yeah, so there is going to be a sort of a number of issues like that in relation to overlapping interests. So if we look at what Manaaki Whenua have done, particularly in relation to at least iwi boundaries, when we look at the sort of the 1,200 items which we've put labels on, you'll see they've still had the biocultural notice on them, because we know that a lot of those are in an overlapping area with us in Tūhoe or us in Aotearoa. And that's actually recognised. So anything that sits in both spaces shows up under both parties kind of, filter.
But do you need agreement from both parties to share the information outside?
No. So this is the difference, because a lot of the information is already being shared and it's already open. And so it's more about how do you acknowledge their interest in the information that's already shared. Now that's a level of activity. I think there's another level of activity, which is a different discussion, which is one around access. And so, there's sort of questions around whether stuff should be open or whether it needs permission to be out there. We have anyway in relation to certain bits of information. Sometimes we decide things are commercially sensitive, so we won't put that in a public domain. There's different bits of information that will be culturally sensitive. Wāhi tapu, things like that, they just want to obscure a little bit. So, there's different ways that we deal with that anyway. This is about ensuring that their name is associated with the information when it's in an open space, so that other people, when they're engaging with it, have the opportunity to engage with the right people.
Yes, That's good. Yeah. Okay. Hey, cheers. Thank you very much.
And just some of the sort of hapu iwi stuff. You know, a lot of that stuff is just sort of internal things that they need to work out. What they can do though with the labels is, you know, if there's an obvious that there's a hapu that is the one that is the authority for a particular space or a particular kind of set of samples, they can indicate that with the labels as well. But what the label does indicate that actually is a hapu. What you do need is some space to then say who that is. Now, whether that's on your system or whether that's kind of internally with the iwi stuff, they can work that out.
Kia ora. Hey, thank you very much. Cheers, guys. Great talk.
Kia ora.
Thanks for the question, Richard. I'm sorry I missed your hand up earlier. So I can see Monique Lads has actually got her hand up. Kia ora, Monique.
Kia ora, Libby. Kia ora, Maui. Thanks so much for that, and thanks for staying on as well. It's really cool. So I work for Department of Conservation, and I look after the monitoring for marine reserves. So thanks, Shaun, for the shout out on our new monitoring framework. It's really cool, and we do want to put more eDNA stuff in there, but as everybody kind of knows, it's quite complicated. So looking forward to seeing this develop. And that's kind of where my question leads. Is it's partially for Maui and partially for everyone else as well, which is there's obviously a big gap in the coastal fishes that you're trying to fill. So we've got resources at place around the country with our marine reserve rangers that are super keen to be involved with stuff like this and to help. So two-part question, which is there a list of things that you would like us to keep an eye out for and help us to develop that? But then Maui, we're really conscious of like how we actually do that in partnership with iwi hapu whānau at place. So if we have that list, is it a conversation with them of saying, hey, are you happy for us to collect these things while we're out and about, or would you like to be involved in the collection, or is there some kind of combination of things that we can do?
I can speak to the first bit. So there certainly is a list. So we had a hui a couple of weeks ago now where we prioritise what are the most important species in terms of those that are most dependent on estuarine habitats or protected fishes, for instance. And of those, we have about 38 that we don't have collections for in New Zealand that we're aware of. So that would involve new collecting events. And the way we've approached that so far, not that we've started because it's early in the program, but we've talked with our museum partners who have usually better established relationships with mana whenua than a lot of other institutions. And we're going to be working with their teams primarily to facilitate those collecting events. But I'm really interested in what Maui has to say, particularly in the marine space where, often these species, you don't even need to apply for any kind of permit for research purposes in some cases if it's not a fisheries species and if it's not, a species that Department of Conservation is particularly interested in, or it's not in a marine reserve. A lot of researchers won't often apply for a permit and then therefore are often not alerted by Department of Conservation or someone similarly that they should be talking to mana whenua. So we'd like to turn that on its head and do it the right way. But yeah, there's no rule book out there. So be interested in your gut feeling on that, Maui.
Yeah, so I think there's as I said, desire for people to engage with, the right sort of mana whenua and communities around these sorts of projects. Sometimes the ability to do that is dependent on the capacity. And that sort of, I think, is, and I guess we don't want to be in a situation where that becomes a block for activities taking place or then things moving forward and people feeling like they've been left behind. And I think this is where, that sort of different sorts of conversations. One's about the actual project itself, that ability to participate in that level of participation, but then what sorts of things are in place within the infrastructure which allow them to come to things later. The project itself doesn't have to be dependent on their capacity to engage, but there's things that they can reflect later on what their how they want their interests to be, recognised, and then their sort of participation in the activities that flow out of it. That's what at least that's what we're trying to do with the infrastructure. And so, but it's an interesting conversation, because I think you get into it a little bit when people start talking about some of the data sovereignty conversations, which is really around kind of next uses and who's got access and where those things sit. And I think having a few tools or options in place make that sort of an easier one to work through than just trying to, work it out yourselves.
Yeah, that makes a lot of sense. And I think the other thing I'm hearing you say is also, even though you've asked Mana Whenua maybe once about, do you want to engage now? They may not have capacity. It doesn't mean you shouldn't ask them again. And that's kind of the approach that we've been taking, is we start with that conversation, and then if it's not timely, then we'll ask them again and we'll say, hey, we've got a little bit further with this project. Are you interested in jumping on in now? So we can definitely integrate those sorts of approaches into doing this sort of thing. But yeah, if you do want us to be involved, let us know.
Yeah, thanks. Yeah, Clinton is on the project team, so yeah, I'll communicate with you and Clinton for sure. Just following on from something I think Maui hinted at is, the notices and labels are one tool. There are other things happening out there. So I know that Cawthron crew have got their own approach for looking after those reference sequences in a way that they're not made publicly accessible by default. And generally in New Zealand, through Genomics Aotearoa, the Aotearoa Genomic Data Repository is potentially a place where for reference sequences for species we think may be particularly sensitive, you can make a judgment call as a researcher potentially, or under the information in conversation with Mana Whenua. That is a repository where genomic data can go that is secure and where access is controlled by nominated kaitiaki. So that is a tool that we have in place in New Zealand currently. And, if it starts there, it may not stay there forever. It may then become public in the domain, but it allows that time potentially to have those important conversations. Yeah, so just for people who may not be aware of that also existing. I don't know if anyone wants to add anything to that conversation or has another question for anyone.